All right, we are going to discuss functional groups. So what are functional groups? So we're looking at these tiny little decorations, and these tiny little decorations are going to be involved in chemical reactions and give organic molecules distinctive properties. And when there is a molecule and it's made up of hydrocarbon backbone, there's going to be a functional group that's attached, and this is that part of the molecule that's going to be involved in the chemical reaction. So we are going to look at hydroxyl, carbonyl, carboxyl, amino, sulfhydryl, phosphate, and also methyl group. First one that I want to discuss is the hydroxyl. So what is it made of? Notice we have oxygen and hydrogen. So typically compounds, organic compounds that have hydroxyl group are going to be alcohols, and one example here would be ethanol. So what's so special and unique about this group is because we have oxygen that's highly electronegative, that is going to give the polar type of property of this functional group. So you can see right here we have ethanol. So this is a 2-carbon compound, and this part is the one that would be reactive. So it would have definitely high affinity to water because you have oxygen, highly electronegative oxygen there interacting with hydrophilic molecules. Another example of a functional group would be carbonyl. So carbonyl is where you have carbon double bonded to oxygen. And I'm going to give you 2 other examples under the same carbonyl umbrella. So we are looking at aldehydes and also ketones. So aldehydes are going to be, so you'll see there's carbon double bonded to an oxygen, but also the same carbon will be bonded to a hydrogen. So this would be an aldehyde. And an example of a ketone would be where you have carbon double bonded to an oxygen. However, that bond between carbon and oxygen is happening in the middle of the molecule. So we find them typically in carbohydrates. So one way to remember, notice ketone has a O here in the middle, so hopefully that helps you remember that C double bonded to O is going to be also in the middle. And then also keep in mind that notice the C, the carbon, is bonded to other carbons instead of the actual hydrogen. So this functional group is also going to be polar because obviously we see the presence of oxygen here. Carboxyl is the one that's going to have carbon double bonded to oxygen. However, there's going to be an extra hydroxyl group. So while we find this type of functional group in acids, and examples would be fatty acids, amino acids, and then what this means is that they can actually release hydrogen. So, and of course, if you release hydrogen into the environment, pH level is going to drop, and we say the number is going to be lower, so it means the environment becomes acidic, more acidic. So we're looking at 2 versions of this carboxyl group. So this would be non-ionized version. You can see the hydrogen is still there, and then if it drops the hydrogen into the environment, now we don't have it here, and you can see since we lost a proton, this is going to be negatively charged end of carboxyl group. So this would be the ionized version of it. So for this reason, the ability to release the hydrogen into the environment allows us to identify this carboxyl group as acidic. So amino group is super easy to identify because we can see the presence of nitrogen, and you're going to see nitrogen is attached to hydrogen. So there are 2, but sometimes you can actually even see 3 hydrogen. So in this case, non-ionized would be NH2, and then ionized version of this amino group would be NH3. So what happened here is the amino group was able to pick up an extra hydrogen from the environment. So if it picks up hydrogen from the environment to form NH3, what is it acting as? That's right, it acts as a base. Another functional group that we want to examine is sulfhydryl. So sulfhydryl is represented by SH, so S stands for sulfur. And I'm going to tell you immediately, you will find that in the proteins. So there's very special amino acid that's called cysteine. And cysteine, and you can see the structure right here. Here is amino group, carboxyl group, and this side chain that has cysteine. That has sulfhydryl in it. So this entire molecule here is called cysteine. So what happens when the protein is folding? 2 cysteines are going to come together, and they are going to form a bridge. So there's another one, and you would have the rest of the amino acid. So this bridge is actually a very strong covalent bond, and is going to be called disulfide bridge. So this bridge, this covalent bond, is going to stabilize the protein structure. And we will discuss the protein structure later, but for right now, just understand that this group, this functional group, is particularly important when it comes to stabilizing and that 3D structure of the protein. How about the phosphate group? So phosphate group contains phosphorus, and phosphorus is surrounded by oxygen, and you can see oxygen at this point are very highly electronegative. Therefore, phosphate groups are extremely reactive because they have a lot of negative charge. Where do we find phosphate groups? In DNA, RNA, also ATP, GTP. You probably are familiar with ATP. This is the energy molecule that can be hydrolyzed to release the energy, and it's synthesized by mitochondria. So ionized form of a phosphate group, you're looking at here, you can see it would, if it was not ionized, you would have H here and H here. And then this is a polar type of functional group. And if you had to identify phosphate group, here it is. You can see shaded blue. All right, and then the last one is the methyl group. So now keep in mind that this type of functional group is not a reactive one. However, it is going to serve as a recognizable tag on biomolecules. To give you an example, methyl is basically CH3. So to give you an example, we have cytosine. So cytosine is one of the bases in the DNA, and then the cytosine can be methylated. So you can see the methyl group is attached to that base. So whenever you have your genes and then some of the cytosine are methylated, that means this part of the gene or part of the genes cannot be expressed. So we say genes that are methylated are silenced. So in other words, methylation can affect the gene expression. So we will have the gene, but it's not going to be turned on. So this one is just a little bit of practice for you to look at different structures and see if you can identify them. So there's one functional group, here's another one, here's another one, here's another one, and this one, if you look at it, it has two, and I'm going to circle all of them for you so you can pause here and practice.